In an ordinary home, a mother waits for a miracle. Her child suffers from a genetic disease, and traditional treatments offer no cure. Doctors tell her that science might change everything. A technique called gene editing could bring hope. But at the same time, people begin to worry. If we start changing genes, are we also changing what it means to be human? Is it salvation, or are we crossing a line? Technology moves forward quietly, while the public walks in uncertainty. In laboratories around the world, scientists are using gene editing tools like CRISPR to precisely modify DNA. This technology allows them to correct mutations, study diseases, and even design treatments that were once impossible. This video takes you into the world of gene editing, to explore its promises, its risks, and what it means for all of us. Gene editing is a revolutionary technology that allows scientists to make precise changes to an organism's DNA. It holds great potential in medicine, agriculture, and even in preventing inherited diseases. The following clip is a short animation sourced from the internet, used here for educational purposes only. It provides a clear visual explanation of how CRISPR gene editing works.
Its basic principles are fairly straightforward, although there are many modifications of this and many layers of additional technologies now being deployed. But at its core, it's a combination of the Cas9 enzyme, which is really good at cutting DNA, and a guide RNA, which directs the Cas9 enzyme to a specific location in the genome.
This guide RNA is designed to be unique to each application. It contains a 20 base pair sequence called a spacer region that's designed to be a homologous base pair match to the cell's DNA. As this whole complex bounces around the cell, the Cas9 attaches to the cell's double helix at short sequences called PAM. The specific PAM sequence depends on the Cas enzyme being used. Each time, this unwinds the double helix just a little bit, allowing the guide RNA to check if there's a match. If not, the complex moves off. But if those base pairs do match, the guide RNA will continue to anneal to the target DNA. Once the Cas9 detects that every base pair of the spacer region is a match to the cell's DNA, it cleaves the double helix. And this by itself can be enough to knock out the gene. Scientists can also provide a repair template to the cell. This is the basic way the technology works, but CRISPR-Cas9 systems are being rapidly developed.
This animation helps us visualize the core mechanism of CRISPR technology. In the next part, we'll explore how it is applied in real-world fields like medicine and agriculture. CRISPR is transforming the way we approach medicine. By targeting and editing faulty genes, scientists can treat inherited diseases at their root, like sickle cell anemia, muscular dystrophy, and even some forms of cancer. Under the microscope, we see the delicate dance of molecules, tiny changes with life-saving potential. For many, this technology represents a new beginning, a pair of wings for those once grounded by genetic fate. In the face of climate change, global food security is under threat. CRISPR offers a powerful solution, enabling scientists to engineer crops that grow faster, resist pests, and survive droughts. These genetic tools reduce the need for harmful pesticides and improve yields in sustainable ways. From golden rice to disease-resistant bananas, gene editing is helping feed the future, one harvest at a time. But how do people feel about editing the very code of life? Let's hear what they have to say.
What are the main problems with gene editing?
Okay, hello everyone, my name is Wang. First, safety, it can cause mistakes and we don't know the long-term impact, so that's a really bigger worry. Then there is ethics. Should we really change genes that can be passed on? It's hard to tell if we are killing a illness or just trying to design people. That could lead to unfairness. Also, many people don't understand this technology, and not everyone has access. That could make health gaps worse.
Okay, so what's your hope for the future?
I hope we use it in safe, far way. If we work together, it can really help people.
Okay, thank you.
Thank you for sharing. Gene editing is growing fast worldwide, bringing big opportunities and challenges. We need to develop its responsibility with care for ethics and human benefit. Countries should work together to share knowledge and close the technology gap. Good rules and global cooperation are key to using this technology safely and fairly. Let's hope gene editing helps build a better future for everyone. See you next time.
Gene editing opens the door to the future full of possibilities, but also responsibilities. As this technology evolves, so must our conversations around ethics, access, and safety. In the end, the power to shape life must be guided not just by science,